On this week's Arabidopsis Research Roundup, I'm joined by Deidre McLachlan, who is working at Bristol University with Alistair Hetherington. And the paper we're going to discuss today was published in Current Biology, and the title of which is "The Breakdown of Stored Triacylglycerols is Required During Light-Induced Stomatal Opening." So thank you very much, Deidre, for joining us today. And if you would, it would be great if you could give us an overview of this paper.
Okay, so we've been working on stomata for a while in this lab, and stomata are very important from plant water use efficiency, crop water use efficiency, because obviously all the water that the plant transpires comes out through the stomata. So we're interested in how the stomata are controlled. They open and close in response to a range of stimuli, and we want to understand this more in an attempt to improve crop water use efficiency. And one thing that's been noticed for many decades now, actually, is these lipid droplets in the cytoplasm of the guard cells. Mm-hmm. And although these observations have been made in many, many different species, nobody really pinned down a function for them yet. So we were interested in seeing whether they might be used as an energy source during stomatal opening. And this was quite interesting because actually we don't know a lot about basic guard cell metabolism. We know quite a lot about function and signalling in response to environmental stimuli, but actually just the basics of guard cell metabolism hasn't really been closely looked at up until now.
Okay.
So we used the neutral lipid stain Nile red to look at Arabidopsis guard cells, and we were able to image these lipid droplets in the cytoplasm of the guard cells. And we wanted to know, obviously, every single morning stomata will open in response to the light stimulus. So we wanted to see if these lipid stores were being used up during light-induced opening in the morning. Mm-hmm. And we were able to see that when we incubated our epidermal strips in the light, obviously the stomata opened, we already know that, but there was also a decrease in the amount of these lipid droplets in the cytoplasm. And to link this in specifically with the light stimulus, we used the phototropin mutant, which we already know doesn't open properly in response to light. Phototropin is the main receptor for the blue light stimulus during morning opening. Mm-hmm. And we saw, as would be expected, that they didn't open in response to light, but also that the amount of lipid droplets in their cytoplasm didn't decrease to the same extent as the wild type.
Okay. Yeah.
So in order to sort of investigate this further, we then decided to look a lot more closely at this pre-dawn to post-dawn transition, and we gathered guard cell enriched fractions immediately pre-dawn and then in post-dawn, and we analysed them for their triacylglycerol content, and we looked at the specific species that were present. And actually, we were able to see that there was a big decrease in specific tag species, and in fact, these 4 species that we saw significant decrease in made up over 60% of the tag species present in these lipid droplets. So this prompted us that, yes, okay, so these lipid droplets are decreasing. It's the most abundant species are decreasing in response to this pre-dawn to post-dawn transition. And we already know quite a lot about lipid catabolism from the work on seeds. So we decided to get some lipid metabolism mutants to see what their stomatal responses were like. And first of all, we concentrated on the lipase mutant, so this is what mobilizes the fatty acids from the lipid droplets. Mm-hmm. And we saw that this mutant had a greatly reduced stomatal opening, and also that there was no decrease in the cytoplasmic lipid droplets during this dark to light transition. So then we went on to concentrate on some of the beta oxidation mutants. So once the fatty acids have been mobilized from the lipid droplets, then they're imported into the peroxisome. And we looked at a couple of the enzymes responsible in this importing into the peroxisome. And we saw a similar thing in that their stomata were not opening in response to the light stimulus to the same extent as the wild type. So there was either delayed or lesser opening in response to the wild type. And in order to put this back more into whole plant context again, we did some conductance experiments on the lipase mutants, and we saw, especially in the first hour from the dark to light transition, that they had much lower stomatal conductance than the wild type in this time. So obviously from a point of view of stomatal opening in response to light, one of the first steps in opening is to activate the proton pump in the plasma membrane, and this acidifies the apoplast. So we decided to look at apoplastic acidification, and we know that this process is an ATP consuming process. And we did this using organ grain indicator in the apoplast microscopically, and we found that almost immediately at the blue light stimulus, the apoplast of the wild type would start to acidify. Whereas with our mutants, this was greatly, greatly delayed by an hour or more. So these mutants, their stomata are not opening in response to the light stimulus properly, and also they're not acidifying their apoplast to the same extent as the wild type. So this led us to think, okay, so these lipid droplets are being consumed, beta oxidation is important in this process, and the energy requiring process, the ATPase at the plasma membrane, isn't functioning properly in these mutants. And just to sort of put this in more of a stomatal energy context, we also looked at the PGM mutant, which is a starch mutant, so it doesn't store starch.
Okay.
And we also saw that in this mutant, stomatal opening was greatly delayed. But also, interestingly, it actually had greater consumption of its cytoplasmic lipid droplets. So to our mind, this was saying that, okay, this mutant is not storing any starch. It can't rely on its starch stores for morning opening, and it is consuming much more of its lipid stores in an effort to open.
I see. So you're seeing in that case that the cell is compensating for the lack of the starch using the lipid as an energy source instead.
That's what we're interpreting this as, yeah.
So really, so thanks very much. That's a really excellent summary of the paper. You went through all the figures nicely in order there, and as you said, it's somewhat unusual to really think about this metabolism of guard cell opening. A lot of the studies are often about ABA signaling or different ions moving into the guard cells for the signaling, but this is a kind of more fundamental aspect of guard cell signaling that you've looked at. So I'm intrigued by this transition at when the lights come on. So is this degradation of the fat stores, does it predict the light coming on, or is that something that you're working on, or is there any circadian element to this regulation?
So we did look a little bit at the circadian elements, and we did see obviously changes over a diurnal cycle in gene expression and also in the amount of lipid droplets in the cytoplasm. But this is definitely one of the areas that we do want to look more closely at in the future.
Okay.
I think really pinning down what's happening before and after the dawn, basically.
Mm-hmm. Excellent. And so I mentioned some hormone regulation. Have you or has anyone else worked on the hormone regulation of this aspect, or is this uncoupled, do you think, from those signaling pathways?
We haven't looked closely at that. No, I mean, obviously this is another area that we would want to look at. We don't know exactly how the light is specifically affecting the lipid droplets yet. We haven't fully integrated this yet. I mean, there is a lot of future work to do, obviously, on this, and to see how this fits in specifically with the metabolism, which I think will be the interesting thing. Metabolism and signalling in response to stimulus have been looked at quite separately up until now. So it'll be really exciting to see how these two things fit together.
Yeah, absolutely. No, we look forward to doing that. So it's I think this area of research is really strong within the UK. So it's good that we have a number of research groups that work on this sort of stomatal signalling area. So yeah, I'm looking forward to seeing more outputs then integrating these signals into some sort of signalling networks. That'll be great. Okay, so thanks very much, Deidre, for giving a rundown of the paper today. and we'll see more production from you in the future, no doubt.
Hopefully, thank you.